and then, okay. And then I'm not going to repeat myself. And then Dian Yang, and I'll let Matt talk about the science aspect of it, but just, it was an incredibly creative process, and where the analysis and our ability to collect and the ambitions and what we could answer with the tool were driven by
sleeping-ish.
What that did and accomplished. But I should say it's also Matt, a great strength of Matt was he was sort of inflappable in the face of adversities. And a theme that seemed to, unfortunate one, I guess, that for Matt's thesis was abandonment. So it started out, Matt was going to rotate in my lab and he was doing great, but he was really computational. We thought he should have a co-advisor. And he had done a rotation in another lab and was computational, was really happy with it. Talked about being co-advisor, and that person went to UCLA. And so then he went back he had this great relationship with Nir from his undergraduate. So Nir was a natural, and as it turned out, was a perfect co-advisor. And then not so far into Matt's PhD, I told him, I was going to be moving across the country, but it was okay because he had Nir in Berkeley. And so, he still had that. And then the pandemic happened, and then Nir decided to outdo me and move not from California to Boston, but to Rehovot in 10-hour time difference. And again, Matt was, sort of inflappable, and he's also very resourceful. And, I'm sure it was lonely at times, and but, I had some reconnaissance there of just how resourceful Matt was when he needed, some advisor or advice, and I wasn't there for him. But we don't really do a thesis defense, but this is, what most places would call a defense. So I wish I could give you this in person. I'll send it when I see you next, but I thought this would be, the perfect thing for your thesis defense. Anyway, with that, I will hand the mantle over to you to talk about science.
Great. Thanks.
Let
me get my screen share here.
Matt, can I have just one or two more words to embarrass you a little bit more?
Oh, yeah, I'm sorry.
Yeah. Do you want me to stop sharing?
Oh, no, you can still share. It's okay. Yeah. So just echoing Jonathan's words on the wonderful experience in working with you, Matt. That was just fantastic. And I was just sitting here and ruminating on this email I received more than 5 years ago from this undergrad at Berkeley who wanted to work in the lab. And after that summer, I think I realized that it's just going to be fantastic to work with you. And there was a really great opportunity. And I guess, as Jonathan mentioned now, right, not a trivial sequence of events, right, that led us all to work together, which I think was just fantastic for me, incredibly productive. And really, Matt, it was, left abandoned and so on, but really you pressed through. You took a lot of responsibility. You even named the project Cassiopeia. Really did a lot of the work that led to this project that is a major project now in the lab, leading the team and so on. So it was a pleasure. I'm very proud of your thesis work. And so let's hear the science a little bit.
Great. Yeah, well, thanks, Nir. Thanks, Jonathan. I'm sorry that you guys tried to get away, but you guys can't get away with Zoom nowadays. It's too bad. But yeah, with anything though, I'll just get started. So this is quite a complex thesis title that I have here. And it's a really hard audience to make a presentation for. We have a lot of different types of people here. We have scientists here. We have people who spend all their days on mountains, and we have some people who are lawyers in this room too, which are probably the most difficult people to present to because they literally hold everything against you. But I'll try nonetheless here to give something that is amenable to everybody in the room. So the thesis title is called Following Tumor Progression Step by Step with CRISPR-Cas9 based single cell lineage tracing technologies and improved computational methods, which is a total mouthful. And to tell you the truth, it's because I normally don't like to talk about it over Thanksgiving dinner. But since you guys are all here anyways, I thought I would kind of dissect this into some more bite-sized pieces. So here's the thesis title. And what I really mean by all of this is what my thesis has been is following cancer from start to finish using really fancy technologies for recording family trees of cells. And new ways of learning these family trees. So hopefully throughout this thesis talk, you can walk away with not only maybe understanding some of the cancer biology that we've started to chip away at, but also some of the cool technologies that we've been able to employ at this task. And we'll see if Thanksgiving dinner goes better this year, I don't know. So to jump in and set the stage to this, I thought I'd roll really far back and just get everyone on the same page of what cancer is. So likely everyone in this room has experienced cancer in one form or another, whether through a loved one or someone you've heard about. And what people have really started to appreciate over the past 100 years or so that we've really started to be able to study this molecularly is that this is a very diverse set of diseases. They're all characterized by a couple of central hallmarks, such as uncontrolled cell growth and eventual systemic spread, the ability to rewire their relationships with the surrounding tissues that they're growing in, and even growing blood vessels within the tumor itself to provide oxygen and resources. But every single tumor is different, whether you find a tumor in the brain or a tumor in the lung. Of course, those types of tumors are different, but even between patients, these tumors are often very different from one another. And compounding on the complexity of this issue is the fact that by the time we are observing the tumor in a patient, it's already grown to this massive mixture of cells that is very complex and difficult to dissect the meaning of these different cells. We see some cells that are able to grow blood vessels very well in a process known as angiogenesis. We see some cells that are able to grow in oxygen-poor environments, some cells that are able to actually leave the primary tissue and travel through the entire body to see distant tumors in a process known as metastasis. But even more interestingly, what we also started to realize is that these cancers can develop from a single aberrant cell over the course of decades, perhaps. And so oftentimes in the clinic, when a tumor is dissected out and we observe the mixture of cells, we don't know all the steps that it took to get to that state that ends up being a danger to the patient. And so we really, I think as a field, are interested in uncovering these steps because they have dramatic ramifications for how you best treat tumors in the clinic. And the most, I think, salient approach nowadays for doing this is with the use of what we call lineages or family trees. So this is what I mean by it. So let's say that we have some tissue of interest, like a lung. A cancer cell emerges somehow by maybe getting some kind of oncogenic mutation that makes it turn bad, and the cell is going to start dividing. And every time a cell divides, it creates two daughter cells. And over the course of this process, repeating over and over and over again, the daughter cells can acquire additional mutations as well. And these mutations, shown as like the orange cell and the green cell right below, can either be good or bad for the tumor. Some mutations allow the tumors to acquire a new ability to metastasize to a different organ, for instance. Some mutations actually are bad for the tumor and cause the tumor to die off. But the important thing to really gather from this kind of process and these principles is that understanding the tumor lineages allows us to really pinpoint exactly when bad things happened or things that made the tumor more aggressive in the process, and hopefully identify weak points or therapeutic levers that we can pull on in the clinic. And So this is a general principle that people have been really appreciating for the past 60, 70 years, the importance of these tumor lineages. And there have been a myriad of ways to try and understand these lineages or try to stitch them together after the fact. And basically all of these lineages, lineage tracing approaches take inspiration from this old fable, Hansel and Gretel, about the two German children who go wandering through the woods and leaving little breadcrumbs by trees. And when they get kidnapped by an evil witch, they're able to escape by following the breadcrumbs back to the forest to come back home. So this is kind of what I mean when it comes to tumors. A single cell will become oncogenically transformed, and as it traverses through the forest to become an advanced tumor, it's leaving, we hope, little breadcrumbs along the way that we can use to retrace its steps and understand the entire cascade of events giving rise to the metastatic and complex tumor that we observe in the clinic. So I hope this general idea kind of makes sense to everybody, and in its conceptual form. But perhaps I can also talk about how this works more definitively in practice. So basically, tumors are growing, let's say, through this metaphorical forest. The idea is we need to find some kind of trees, and the breadcrumbs are left by trees. In the traditional examples of tumor lineage studies, the trees have been positioned in the genome of cells, and the breadcrumbs have been natural mutations that get left over time, either from DNA damage or through the error-prone process of DNA replication. And so if we take a tumor's tissue, for instance, from a lung once again, and we sequence the genomes of cells, we can align these genomes to one another. We can essentially align the trees with one another and look at where breadcrumbs are left along the way. And these breadcrumbs, like I said, are in the form of mutations. And these mutations are highlighted in red here. And so the idea is we can use some sophisticated algorithm to reorder the samples with one another to retrace how the breadcrumbs must have been left. So for instance, it makes sense that all of the cells or all the samples with the mutation, the T mutation in the first position, belong with one another, as well as all the cells on the bottom all have that mutation G in the fourth position, or fifth position rather. And this tree-like structure right here is a family tree. It's linking the cells to one another based off of how the breadcrumbs were laid throughout this process. So this general principle has been incredibly important in helping us understand clinical samples from human patients from myriad of different types of cancers. There's large consortiums that have been developed for the purposes of studying how these tumors evolved and metastasized throughout the body of human patients. But there's a couple of drawbacks that are intrinsic to this type of system. So the first is that we can't control how often DNA mutations are happening because we're relying on natural processes. Sometimes samples don't even have enough mutations to relate one another. There weren't breadcrumbs that were left along the way. Secondly, it's often very expensive to sequence every cell's genome in its entirety. So you can imagine in this type of system, you have to check every single tree if there's a breadcrumb or not. And because these cells' genomes are quite large and you want to be able to study several different cells and relate them to one another, it's difficult to manage to pay over and over and over again to support these types of efforts. Thirdly, we often don't have the luxury to wait a very long time for these processes to unfold. Even if we did have an unlimited budget, even if we did have good breadcrumbs left along the way, we don't often have the luxury to wait decades. Instead, we would rather use something more tractable, something that resembles the physiological process of cancer development, but is a little bit more manageable in the lab. So the ideal scenario here is that we would have exquisite control over when mutations happened, or where we could, how often we're putting down breadcrumbs. The second thing we'd like to have is a relatively cheap way to study these breadcrumbs, or really only look at a specific part of the forest or the trees. And third, we would like to engineer these things into experimental models that are workhorses in the lab for studying tumor biology. So this is kind of where our project took off. And over about 5 years of a fantastic postdoc by Michelle Chan, we developed a technology that allows us to do this. And the technology essentially works by shrinking the set of trees that we need to look at to retrace the tumor's path of the forest. And it uses a very flexible molecule known as Cas9 to install these breadcrumbs over time at the right cadence. So let me define a little bit more clearly what exactly I mean by this Cas9 breadcrumb mechanism and the specific set of trees. So we're using a very basic property of Cas9, which is that this molecule that you might have all heard of as this kind of like wonder therapy for writing new genomic information, what it really is doing most often is targeting a very specific site in the DNA and inducing a double-stranded break. So we can programmably direct Cas9 to a specific target. It's going to induce this double-stranded break, so cut open the DNA and then flee the scene. And DNA will hastily try and repair itself. In the process of doing so, introduce a scar in the form of random nucleotides inserted into the position or just deleting a large segment of the nucleotides. And so the scar is in the form of insertions or deletions or indels, abbreviated as indels. And these indels persist in the genomes over time, just in the same way that mutations were persisting in the tumor example I showed before. So it's a very basic mechanism for writing random information at a very specific site. We took this property of the system, engineered our own synthetic set of trees. And we call these synthetic set of trees our target sites. And there are 3 trees or 3 sites in tandem with one another, each targeted by Cas9 and each capable of acquiring breadcrumbs or indels over time. And we can put several versions of this into a cell to increase the information capacity and the redundancy of the system. So just doing some back of the envelope math about the number of trees and the number of possible indels that we could have at a specific site, we in theory could nuclear mark every single cell in the human body. So if everything was working properly, we wouldn't just be able to trace tumors, we'd be able to trace embryos of mice or some other experimental system as they're growing into fully fledged organisms. And so in practice, how this works is we'll put all this machinery into cells. So shown right here is just a single cell that we have put all machinery into, our Cas9 molecule for leaving breadcrumbs, our target sites for acquiring the breadcrumbs. And as these cells divide, we'll strategically lay down these breadcrumbs, giving us an idea of who's related to whom. And these are heritable, so they get passed on from generation to generation as cells divide, and they start to accumulate. And at the end of an experiment, we will sequence our cells. And even though we won't have observed every single cell division over time, just from this one observation point at the very end, we're able to stitch together how the breadcrumbs were laid down and how these cells were related to whom. And the way we do this is by looking at the set of mutations that these cells have. And so you can see that cells that share the orange mutation on the left-hand side of these strips of DNA are related to one another. And the cells that share the set of blue mutations on the far right are related to one another as well. So the same property that I was telling you about from tumor phylogenetics, it's just now we have a more experimental system for doing this that's more tractable. So the other great thing about the system that we're able to exploit is that we can read this information off of mRNA, which gives us not only an idea of how cells are behaving, so if cells are good at spreading throughout the system or spreading throughout the body, or if cells are good at growing blood vessels, for instance, but we also get the lineage information as well. And we normally summarize the lineage information in the form of these character matrices, which is just computer science parlance for the mutations that a cell has, the breadcrumbs that a cell has. And we align each one of these rows or samples with one another with respect to the trees. And each unique color here is a mutation or a breadcrumb that we can use to stitch together the relationships between cells and come up with the trajectory that the cells took. Now, up until this point, I have intentionally been pretty hand-wavy about how we actually uncover the relationships between cells. But in reality, this is a really difficult problem and a problem that we thought about for a long time. And the reason why this is difficult is that it's what's known as an NP-hard problem, which once again is just computer science parlance for saying it's not trivial to solve, and you really have to spend a lot of time testing out complex rearrangements of the data to find the best solution. And the reason why this is because of this phenomenon known as homoplacy. And homoplacy is essentially the phenomenon by which breadcrumbs that look identical to one another get left on two separate trees. And this is a problem because if you're not careful, you might be tempted to think that the breadcrumbs are the same. But in reality, they're different. And so if you have the ground truth at your disposal, you can see that two, for instance, orange mutations were left at separate parts of the growing cell population. But if you just observe the bottom of the tree, you'd be tempted to put the cells together that are all orange. So this is generally difficult. This is common in the regular systems that I was telling you about before. But in our CRISPR-Cas9 system, there's a couple of more difficult difficult challenges we need to overcome. First is the fact that we're dealing with a large input size. So we often have way more samples to process simultaneously than previous applications of tumor phylogenetics or tumor lineage tracing. So that means that not only is this problem difficult to solve, but also we need to have efficient algorithms for doing this. Secondly, we often don't get to observe every single one of the trees in our system. We don't get to observe all of the sites. And so we also have a lot of missing data that we need to be able to be resilient to. And lastly, since we're using these indels, these insertions and deletions as our breadcrumbs, this means that it's non-traditional, that we have to come up with new models for handling the indels, but then also certain indels are much more common than others. So certain insertions, for instance, are pop up as the outcome of the Cas9 operation way more often than other types of indels. And so this actually increases the chance of two identical looking breadcrumbs being left at separate trees, or increasing the chance of homoplacy. So in work that spanned about 3 years and got published about 2 years ago, I'm just going to distill down to a single slide because it's quite complex. The idea is that we came up with algorithms that could exploit basic kind of assumptions about the data to improve the scalability of the solution, but at the expense of not finding the most optimal solution. We call this a greedy algorithm because it's greedily taking different types of solutions at the expense of making small errors along the way. And we also came up with solutions or algorithms for solving this problem that are more complex, more difficult to run on larger samples, but are guaranteed to give optimal results about how cells are related to one another. And we also can marry the merits of these two different algorithms in a hybrid algorithm called Cassiopeia hybrid. And so suffice it to say that we have at our disposal a computational toolkit that is adaptable to the type of problem that we're given. On very large data sets, we can run this greedy algorithm. On very small data sets, we can run this more precise algorithm. And on the intermediately sized data sets, we can run a hybrid algorithm. So at this point, we have an suite of algorithms that we could deploy, and we have a system that theoretically is capable of recording very large lineages and allowing us to stitch together the path that a tumor took throughout its course. The first proof of principle that emerged was published last year in collaboration with a fantastic postdoc named Jeff Quinn. And the proof of principle that we took was a cancer cell line, known as A549 cell line. They're derived from a human patient with non-small cell lung cancer, and are kind of a workhorse system for studying metastatic tumor biology. And we could take these cells, we could put inside of them the lineage tracer system that I was describing before, so the Cas9 molecule, our synthetic target sites or our trees, as well as some fluorescent markers of the cells that we could use to basically trace from afar how profuse the metastatic spread was in a mouse. We took about 5,000 of these cells, injected them into the left lung of a mouse, and over the course of about 2.5 months, we allowed these cells to metastasize throughout not only the lungs, but also the liver, some lymph tissue as well. And shown right here on the bottom left is actually the fluorescent signal that we could acquire as the cells are metastasizing throughout the mouse. So you can see that on the leftmost panel, the signal is quite local to the left lung, but then as time goes on, you can see it spread throughout the mouse. We can sequence the cells after dissociating the tumors, and we sequence about 41,000 cells from 6 major anatomical sites. We sequence cells from the left lung, which is the primary tissue that we introduced the cells into, as well as the right lung mediastinum, which is lymph tissue between the lungs, as well as the liver. And as promised, we have a lot of information that we can get for just a single subset of the data or a single clone of the data. So this is showing about 51 or 5,500 cells that were acquired from this tumor model. We have about 51 synthetic trees that we put into the system that were acquiring breadcrumbs along the way. And we have uniquely marked basically every single one of the cells. So what this essentially means is that we've engineered in such a way at the right cadence of putting down breadcrumbs that we could follow each cell trajectory back to the very beginning. So to walk you through a little bit more of what this visualization is showing, on the left-hand side, that spindly black web, that's the family tree of cells. The rows of this tree each resemble, or each correspond to a single cell or a single sample in our system. And the columns are the actual sites that were being mutated over time, so the trees in the system. The colors are the mutations that the cells had. And so if you just take a very far back view and you kind of squint and you look at this, maybe some of the patterns that emerge right off the bat are that cells that have similar profiles are related right next to one another. So there's like this blue strip down the middle. Hope you can see. All the cells that have that same mutation are put right next to one another. So this is kind of a good sanity check to start with. But if we zoom in to even a small part of the tree, just about 500 cells or so, we can see that the information doesn't shrink in complexity. It pretty much stays the same. It's quite fractal in nature. And we can get even more of an appreciation for how cells with similar mutation profiles are being put next to one another in this tree. And this remains true even if you zoom in even more to a smaller part of the tree. So, and we use the Cassiopeia hybrid algorithm here. And so hopefully you can appreciate how I was able to scale and also return a solution that makes sense just by eye. So with this type of complex data set and the algorithms at hand and the trees that we're able to reconstruct, the really fascinating thing that we're able to do with this system is retrace how metastasis happened throughout the body. What are the routes a cell took from the left lung to a different tissue? And let me walk you through exactly how we're able to do this. So shown right here is just a family tree of a small subset of cells. These cells are now annotated by which tissue the cells came from. And what we're able to do is just use basic logical properties to reconstruct how the cells spread. So for instance, we can infer that that cells started in the left lung and moved to the lymph tissue known as the mediastinum early on in the experiment. From there, we can find several examples of cells moving between lymph tissues in the mediastinum. We can also identify cells moving from the mediastinum to the liver, as well as cells moving from the mediastinum to the right lungs. And the important thing to convey here is that this is the first time we've been ever able to explore the metastatic routes that cells took at this kind of resolution. And we found that basically cells took very erratic roles throughout the mouse. There wasn't any distinct topology that was reproduced over time. And I think really shed a lot of light over late stage metastatic disease. So we were really proud of this work. It was a great showcase for the technology, and I think shed some light about how metastasis was happening in this experimental model. But recall what I said before, that cancer is a long process starting from the single transformed cell to this late stage tumor metastasis. And the model that I was just talking to you about before really only explores one part of this. It's only exploring the metastatic part of this cascade. And it doesn't really shed any light on what happened up until that point. So ideally, we'd like to kind of take the curtain back a little bit and look at this entire process. And in order to do that, we need to have a way of initiating a tumor from its very beginning. And we need to have a way of laying down our breadcrumbs from the very beginning as well. And so in the last part of this talk, scientific talk, I wanted to describe where we're actually able to do this. So in collaboration with Deanne Yang, another postdoc in Jonathan's lab, we engineered a genetically engineered mouse. So the genetically engineered mouse carried two oncogenic mutations, KRAS and P53, that are often mutated in patients with non-small cell lung cancer. And when put into a mouse, is a great model for the progression of non-small cell lung cancer. Now, under native conditions, the oncogenic mutations are not active. So this mouse grows up just fine. But upon the introduction of lentivirus particles, we can actually induce the tumors. And simultaneously, we can also induce the tracing inside of the tumors as well. So from the single cell progenitor of cancers, we can actually start laying down breadcrumbs as it starts to grow into a tumor. After about 4.5 months, we can take out the tumors, we can dissociate them and sequence them and stitch together the entire biography of how the tumors were growing over time. So a couple of just short vignettes, I want to tell you what we can learn from this type of data and why we're so excited about this platform. The first thing that we learned about this system is that particular subsets of the tumors are a lot better at growing than other subsets of the tumor. So essentially, if you imagine this linear progression from single transformed cell to complex tumor, one of the first thing we found is that certain cells go faster through this trajectory than others. So I'm highlighting in red here on this little family tree is a subset of the cells that are expanding or growing faster. And we can actually quantify this using a statistic called fitness. So how fit the cells are in the tumor itself. And when we correlate this with the expression of specific genes that we get from the sequencing assay, we can actually identify genes that are associated with cells that are growing faster and genes that are associated with cells that are growing slower. We did this across all of our tumors to come up with a signature of reproducible genes that are considerably making the cells grow faster or slower. And when we export the signature and apply it to a data set of human patients with lung cancer, what we found is that this was actually significantly predictive of patient survival. So shown right here is what we call a survival curve. And so every point on here is a patient who's deceased. And the blue curve shows patients who are stratified as not expressing the genes that we found associated with fitness. And you can see that those patients tend to survive longer. And in the red curve, these are patients that do express the genes that were associated with more fit cells in our tumors. And these patients die sooner. And so while it's a bit of a morbid thing to be excited about, we actually found this to be quite encouraging that we're able to stratify patients in such a way and indicating that we have clinical importance in our assay. The second thing that we were really excited to find is that not only did certain cell subsets grow faster, but there's also an important bifurcation in types of tumors along this trajectory from single cell that's been transformed to metastatic tumor. So what we found is that along the way, tumors choose between one of two paths. They either become more gastric-like, expressing gene programs that you find in stomach cells, for instance, or they become resembling of more progenitor-like lung cells, so expressing genes that are expressed early in embryonic development that are that lead to this to lung tissue. But regardless of which bifurcation or which adventure that they choose, they each land on this advanced tumor genetic state and capable of metastasizing. And this, to us, was extremely exciting because with this assay and this technology for tracing putting breadcrumbs along the trajectory, we're able to deconvolve whether or not these two different types of important states were related to one another or completely separate from one another. And the fact that they're completely separate from one another, I think, sheds a lot of light on other types of tumors, perhaps being able to exploit similar mechanisms of choosing different adventures, so to speak, along their trajectory. Building on this kind of idea of different kinds of paths to the forest that tumors can take to the advanced state, we also knocked out additional tumor suppressor genes that are commonly mutated in human lung cancer patients, such as LKB1 and APC, and we found that we could completely reorient the path that tumors took through the forest. So this, I think, has two major implications. The first one is that it's much more of a complex story than we originally realized, that not only are tumors different between patients, but depending on the different genetic backgrounds, you could have completely different routes that tumors take to the advanced stage. But even more importantly, this system, because we can multiplex genetic perturbations on top of this tracing system, we're able to really, in high throughput, explore how tumors are evolving. So the kind of promises that this gives is you might be able to take a genetic profile from a patient that you learned from bulk sequencing of the DNA, import into this mouse model, and study how the tumors are evolving in situ over time, and hopefully shed light about how these tumors might be able to be treated in the clinic better. So this really wraps up the scientific part of my talk. And just to put it all together, I've told you about our fancy technologies for recording family trees using this Cas9 molecule to write down information at these specific sites. Or in the Hansel and Gretel analogy, we could put down breadcrumbs that are specific trees, allows us to uncover paths of the forest. We had better ways of reconstructing these family trees that met the demands of this type of technology. And using these tools, we're able to shed light about the topologies or the routes that, that cells took throughout the body of a mouse for the first time ever. And we're also able to reconstruct the evolutionary trajectories that cells took as well, from the very start, the inception of cancer, to the advanced histology that you observe in the clinic. So it's been a magnificent scientific journey, and hopefully I've conveyed just an ounce of the excitement that I feel for this, and also hopefully give you kind of an idea of the kind of things that I've been working on for the past couple of years. At this point, as Jonathan said before, it's not really much of a defense, more of an exit talk and a celebration. So I want to spend a considerable amount of time going through the acknowledgements of all the people that made this possible throughout along the way. So first and foremost, I want to thank my advisors. Jonathan has been just a fantastic person to work with. He met me when I was fresh in the PhD program. And I had no real experimental background. I was extremely naive about the types of things that were possible, but he took a chance anyways on someone who could code and stuff. And it's just been a fantastic scientific journey, learning under Jonathan how to approach science and how to weave together a story and how to build something that's robust, and as well as put on your detective hat and figure out what exactly is giving rise to interesting phenomenon. As he mentioned before, I've been quite resourceful in my PhD, but also he's been quite busy. So we've had to use, like he said, approaches for accommodating his busy schedule. And so here we show this picture, but this is Dan Santos, a PhD graduate student in the lab, wearing his face. Not nearly as cool as when Hannibal Lecter did it, but it's still cool anyways. But we knew that he was having a good time no matter where he was. And you could always rely when you were with him, he was going to be a good time as well. So this is him playing his favorite game, Flip Cup, at one of the retreats. And Nir as well. You know, like you said, you've known me for the past 7 years. And I attribute all of my success virtually in getting started in my research field to you just taking the chance and on me and joining your lab. It's been fantastic to work with you. You have been extremely patient with me. You've been extremely creative with me as well, thinking about the best ways to solve these types of problems. You've given an endless fount of ideas. And I'm also incredibly appreciative that you've made this long distance advising situation work as well. I have the better end of the deal. For people on this call right now, you probably don't appreciate how late it is for Nir. It's 10 p.m. for you right now or something. And he does this every single week in, week out for the past year, advising his students. So he's an incredibly devoted advisor. And I'm just so lucky that I got to work with him over the past couple of years. As you might know, or might expect, he's also extremely busy. And for those of you who have been in the lab long enough, you've probably received a series of these emails of nondescript subject lines with no message body whatsoever, just saying status, updates. Well, status of what, Nir? Updates of what? But he's a busy guy. What can you say? But we know that we'll eventually track him down, probably on Twitter, and know that he was also having a good time with his colleagues, and we could rely on him having a good time when he was with us as well. So I'm incredibly fortunate to have had advisors who've cultivated good lab environments as well. Over the years, there's been a lot of turnover. That's just how these PhD programs work. Every couple of years, there's a new cohort of students and postdocs. And just over the years, I've been just so fortunate to work with fantastic scientific collaborators. In 2019, Jonathan generously took us to Hawaii, and we really bonded over science and snorkeling and everything in between. This is probably the best photo resembling the first wave of scientists that I got to work with in Jonathan's lab. Some of them still around as well. Nir also generously always dished out a fantastic barbecue every single year. There was a small hiatus placed during COVID, but we got together last June for our summer barbecue in a park. And it was great to see the faces that I missed for so long. And hopefully some of them are on this call as well. And I just want to let you know, thank you for fantastic scientific discussions and support and laughs all along the way. And the Cambridge lab has really grown into a fantastic ecosystem for doing science. It's like a biotech party every time I go. Everyone's so excited to talk about science, and also go out to dinner and not talk about science as well. So this is just from a couple months ago, we went out to dinner in celebration of me graduating a little early, but it's just been great to get to know people, even just virtually, and collaborate and with them. So just thank you so much for being such a welcoming group of people, absorbing in an old dog who doesn't really visit all that often. And I'm so appreciative of all your support. A subset of the labs have been focused on lineage tracing as well. And so I want to just thank specifically some people who have made this journey especially fruitful. Deanne has been a fantastic collaborator. He's been also extremely patient with me and been very patient through a long distance collaboration relationship. We've made it work, even if it meant waking up every day at 8 a.m. to have research calls and write papers together. I really hope that we get to collaborate more in the future, because it's rare that you find such a fantastic collaborator that complements one another. Jeff Quinn was the person who really brought me onto the team and really took me under his wing. He even took the chance to teach me a couple of things at the bench, which was really dangerous on his part, because I'm pretty sure he had liability if I hurt anybody, but luckily I didn't hurt anybody and it was totally fine. But he was extremely generous with his time once again, and extremely diligent. He would look at my analysis under he'd pull up on a big screen and look at the actual sequencing reads, and go through with a fine-tooth comb to look at aberrations and stuff. And I had a lot of bugs in my code starting out, and he was extremely patient finding them. So thank you to Jeff. And Michelle was the pioneer of this technology, and I was so fortunate to get to overlap with her for 2 years before she started her lab at Princeton. And if Michelle's on this call, I just want to say thank you for bringing me on and being once again so generous with the technology and making room for me on the team. And I still bug her all the time with questions. So it's one of these relationships that, for better or for worse, Michelle, you're not going to be able to get rid of me. Also, grad students as well, I got to work with on a lot of the analysis side of things. Alex was my partner in crime, developing Cassiopeia. Sebastian and Joseph are new additions to the team who, I've been so happy with how they've been taking a lot of the directions as I start to move on to my next position. So thank you for putting up with me, but also giving me new ideas and making me a better scientist along the way. I also somehow convinced several people to work under me as well along the way. I got to advise several master students and research technicians and undergrads, especially through Berkeley. A couple of them stuck around for a really long time. Richard was with us for about 3 years before starting his position at Princeton. And Yunai is now at Stanford, and Halil has got a great job in France. And I could go on and on about each one of these. But I always say to them that there's nothing better in science than working with one of these people that are under you and watching them outgrow you and move on to a new stage and helping them get there. And so I found it to be incredibly rewarding. And for any of them on the call, thank you so much for letting me learn how to be a better mentor and being patient with me as well. It's been just a pleasure to publish with you, to work with you along the way. Also was incredibly fortunate to have a great thesis committee Matt Spitzer and Hani Goodarzi have been with me from the very beginning of my PhD, way before their obligations as Thesis co-committee members started. Matt was helping us brainstorm stuff about the KP mouse with Deanne from the very beginning of my PhD. And Hani, I met at a pizza party, maybe like my first month of my PhD, and he's just been a stalwart of support and just a staunch staunch supporter of me throughout my entire time. And I know I can turn to either one of these with career questions or personal questions about how to manage things going on in my career, and they will jump at the opportunity to help me. And so I've just been so fortunate to have these people in my life as I pursued this PhD, and I'm certain I will stay in touch with them. So Hani and Matt, thank you so much for your time and patience and generosity throughout the way. So besides science, believe it or not, I also have a life outside of science as well, and that has also made it incredibly rich over the years. I have friends spanning from early in my elementary school days to today that I still stay in touch with, and they just make life worth living a lot of times. A lot of them are on this call and doing way cooler things than I'm doing, and so I hope one day I can go to an exit talk for you guys in some form or another. But just thank you to people who've been with me along the way, backpacking or over family dinners and stuff like that, who bring color into my life. So thank you so much to those people. I also obviously wouldn't be here without my family, quite literally. And so I wanted to thank first my biological family, who managed to provision me with a couple of brain cells to do this research. And in all seriousness, Mom and Dad, I'm so thankful for your support in me, your patience as well of trying to learn what I'm trying to do. This isn't the first time I've tried to tell you about my science. I really hope this came through a lot more clearly than the past times I've tried to tell you about it. But I knew that you guys would be an ear to my problems and just showing me so much unconditional love and support. And to my brother as well, who's a younger brother, but I honestly consider a big role model, the way that he handles himself with other people, the way he respects other people, and the way he treats me as well. And you can imagine being a big brother and little brother growing up, there's a lot of reason to have animosity over the years, but I'm just so glad that we get along, and that also he works at a winery, so we get a lot of wine from him as well. So thanks, Jack. My also in-law family has been just fantastic as well. I've been with my wife, and you'll hear about her later, for about 12 years. So it's really a second family in every aspect of that word. They've seen me grow up since I was a kid, pretty much in high school, even before high school, to who I am today. And so they really, I think, have shown also a lot of patience, just being patient with me in grad school, and knowing that one day I'll get a real job, and one day I'll make money, and I'll be fine and independent. But they've just also been eager also to hear about my work, and a constant fount of support as well. And also to Aubrey's sister, Kara, has been a fantastic sister as well, also incredibly supportive, and her very significant boyfriend, Matan, has also been a fantastic brother to have as well. And they've both been in Cambridge too over the past couple of years. So visiting Cambridge wasn't just about doing science, but also visiting you guys. So thank you so much. And last, but certainly not least, my wife, Aubrey. So hopefully many of you have met her by this point. And if you have, you'll know how generous she is, how patient she is, and how just kind she is. And once again, she's put up with this whole PhD journey for a very long time as well, through all the ups and downs, and has made it incredibly worthwhile to do. We recently just did the Community Santiago together, and so I'm also very appreciative that she took many weeks off from her job to travel with me. And so it's trite to just say I love you, but it's so true. And this has just been this journey would have been impossible without the support and love that you showed me over the years. So thank you from the bottom of my heart, just for being a fantastic wife. And where I'm going next, I also have a small thank you to the people taking a bet on me next. So I'll be going to Stanford to pursue postdoctoral work in Howard Chang's lab. And I would be a very poor Cal graduate if I didn't choose the seal with the junior university in there. So despite the fact that it is the Leland Stanford Junior University, it's a pretty great place to be, and I'm really excited to be there. So with all that, thank you so much to everyone tuning in today. And if there's time for questions that you're interested in, I'll be happy to take them. Sorry, thanks, Luke. Not, yeah.
All right. So what are you excited about doing next?
So yeah, I'll be continuing thinking about tumor evolution, which is exciting scientifically. We'll be staying in the Bay, which I'm also excited about. So if anyone is in the crown jewel of Silicon Valley, San Jose, do let us know. Yeah, but working with Howard is going to be a lot of fun. And thank you to Neer and Jonathan for obviously providing good letters of recommendation for me to get this job.
You left us speechless.
There you go. That's, I guess, the goal. Well, you know where I live, most of you at least.
And you have to come back sometime.
I'll be there.
I will. So people might have noticed I was not at that dinner. I was sequestering at home with potential COVID at that time. So I have to come back and do a proper celebration again.
I'll take you up on it.
Final round of applause.
Yeah.
Thanks. All right.
Everyone.
Well, you know, let me know if you have any